Am I audible to you? Just confirm me. Am I audible and the PPT is visible? Okay, fine. In today's class, we are going to discuss about mutation. Okay, and before we'll discuss about the topics, just I want to make sure that you have been very well with the concept. Okay, so what is mutation? Generally, mutation means a sudden change. Okay, a sudden change, and that mutation will leads to a positive impact or even a negative impact. So we can categorize these mutation into three different types. One is gene mutation, chromosomal mutation, and genomatic mutation. In gene mutation, we have further two different types, frame shift mutation and substitution mutation. Frame shift mutation means deletion. Nucleotides, we have DNA, and DNA is made up of nucleotides. Okay, in that nucleotides, if some of the nitrogenous bases have been deleted, that have been coming under the category deletion. And if some nucleotides will add or insert in the middle unknowingly, and that will be calling as the insertion. So these deletion and addition or insertion will be coming under the frame shift mutation. Why? Because if it is a deletion and if it is an insertion, anyhow the nucleotides will be vary. Okay, as a result of this deletion and insertion, the nucleotides, the amount of nucleotides will vary. The entire frame will get mutated. So that is why we are calling them as the frame shift mutation. So in the substitution mutation, we have a transition and transversion. What is transition and transversion? And if a purine is replaced by another purine, and if a purine is replaced by either purine or pyrimidine, based on the mutation occurs in purine or pyrimidine. Okay, that is why we are kept under the substitution mutation. Let me explain in detail in the upcoming slide. So next, the chromosomal aberration. Chromosomal aberration means for the deletion, if some of the nucleotides has been deleted, that is called deletion. Duplication, if the nucleotides will be duplicated, for example ATGC, and that ATGC will be duplicated once again, that is what a duplication. And translocation means it has been relocated. Okay, the place has been changed as a result of this translocation. Inversion means, for example ATGC means CGTA, the entire frame has been inverted. That is why we are calling them as the inversion. And further, this inversion has been categorized into two types. One is pericentric and paracentric. Whether the centrosome, whether the centromere will be involving or not. Okay, and if the mutation occurs between the, yeah, that if the centriole or the centromere, if the centromere is including or not, based on the position of centromere, we are categorized them into a pericentric and paracentric. Okay, and in genomatic mutation, we have aneuploidy and euploidy. In aneuploidy, we have a trisomy and monosomy, and in euploidy, we have a haploidy, diploidy, and polyploidy. And this polyploidy will be further categorized into autopolyploidy and allopolyploidy. See, we are going to discuss this first half in this class and the second half in the next topic. There is chromosomal aberrations. Okay, chromosomal disorders. In that topic, we are going to discuss all these things. For now, we are going to discuss only the first part. So, once again, what does frame shift mutation means? If the deletion is happen or if any addition of nucleotides or insertion of nucleotides, then the entire frame will get mutated, entire frame will get modified. Only then we are calling them as the frame shift mutation. And here we have a frame shift insertion, frame shift deletion, frame shift duplication, and frame shift insertion and deletion. Okay, and here based on the substitution type, in the substitution mutation we have a different types of a mutation. When a single nucleotide substitution, here only one nucleotide has been replaced by another nucleotide. Okay, and in silent mutation, here if any codon, for example, one codon for AUG is code for a methionine, and some other codon will code for the serine. Okay, as we said before, many nucleotides can code for many codon can code for a same amino acid. Here, one codon is code for the serine. Also, a different codon can also able to code for this serine. So, anyhow the amino acid code for this particular codon is not going to change, but anyhow the codon here will be changed. That is what a silent mutation. Here there will be a no impact will be present. Okay, only a minor impact will be present. And missense mutation, which means what? The codon codes for a different amino acid. If a different amino acid will be present, the protein synthesis will be vary. That's what a missense mutation. Nonsense mutation means here a codon changes to a stop codon. Okay, we know that a protein synthesis will be stopped when the tRNA reaches the stop codon, because there is no amino acid can able to code for this particular stop codon. So in the middle itself, if the codon is converted into a stop codon, means the protein synthesis will not occur. This will be the dangerous mutation than the above three. Okay, and this is the most dangerous mutation, and this will be the minor mutation, that is silent mutation. Okay, and here a transition and transversion. Here the purine nucleotide will be code for another purine. Okay, for example, adenine is a purine, guanine is a purine. Here this adenine purine is replaced by a guanine. In that case, we are calling them as the transition. In case it is a pyrimidine nucleotide, and that nucleotide will be mutated to another pyrimidine. Okay, that is what a transition is. But in case of transversion, a substitution of purine nucleotide for a pyrimidine or vice versa, which means what? A purine, for example, this is a adenine and guanine is a purine. Here in transition, this adenine is replaced by a guanine, and guanine is replaced by an adenine. And thymine and cytosine is a pyrimidine. Here, thymine is replaced by a cytosine, and cytosine is replaced by a thymine. This is what a transition is. Only 4 combination is possible in case of transition. But in transversion, see ATGC, adenine can code for a thymine, guanine, cytosine. All the way is possible, because here a purine nucleotide can code for a purine or even a pyrimidine. For a pyrimidine, it can code for a purine or a pyrimidine. Okay. So here 8 combinations will be possible in the transversion, and in the transversion only 4 combination is possible. This is what a type of substitution mutation. And one of the most important thing you have to remember is what is loss of function mutation and what is gain of function mutation. Here in case of loss of function mutation, it will be happen. See, we have an allele concept, right? For example, this is height, tall gene. This TT, capital TT, will code for a dominant character and results in a tall plant. Okay. For example, it is heterozygous. Okay, it will be heterozygous. Once again, it is code for a tall. Okay, is it not? And this will be a recessive character. Both are small, and this will be recessive character. This will be code for a dwarf reaction. See here in loss of function mutation, both the both allele has to be mutated. For example, if the mutation occurs in the small t, let's take this tall plant, a dominant character. If the mutation occurs in this T, it is not sufficient. These both allele has to be mutated. Only then this loss of function mutation has to be happen. For example, see in the gain of mutation, even if one allele is getting mutated, it results in a bigger change. In loss of function mutation means both the allele wants to be get mutated. For example, tumor suppressor gene. In tumor suppressor gene, what is the function of tumor suppressor gene? This gene has to suppress the cancer producing genes. Okay, this gene is responsible for suppressing the cancer producing genes. In this particular gene, both the allele has to be mutated. Only then this tumor suppressor gene will lose its function. This is what a loss of function mutation. In case of a gain of function mutation, here it is the best example will be the oncogene. Oncogene means the gene which is responsible for causing a cancer. Here, either of an allele, any one of an allele, if it gets mutated, it will be results in the gain of function mutation. It will result in a mutation. Okay, this is what a difference between a loss of function mutation and a gain of function mutation. Now, once again, the same thing will be repeating here. And a spontaneous mutation and induced mutation. Here, this mutation will be occur spontaneously, that is naturally. That means if there is any problem in the DNA replication, if any problem done by the DNA polymerase, it results in the mutation. If there is any problem in the repair, if if the DNA polymerase did some mistake, means it has to be repaired. If the repair is not occurred, once again it results in the mutation or in the recombination process. Okay, this is what a spontaneous mutation. Here, transition, transversion, deletion, insertion. All the type of mutation can be seen here in case of spontaneous mutation. In induced mutation, it has to be induced by some external factors like chemicals, UV rays, that is radiation, and certain carcinogenic agents, that is cancer causing agents. If these agents tends to induce this mutation means, only then we can call them as the induced mutation. All the biological agents, transposons, that is jumping genes, radiation, UV light, X-rays and gamma rays, the chemical mutagens, that is chemicals and carcinogenic agents. It has been an external factor which is going to induce the mutation. And the spontaneous mutation, there is no external factor. It has been occurred by themselves. Okay, this is what a difference between spontaneous mutation and induced mutation. So the first question, what type of mutation involves the addition or deletion of nucleotides? Okay, that will be the frame shift mutation. Just now I have been explained, if there is any addition or insertion, or if there is a deletion, that will the entire frame will get modified. That results in a frame shift mutation. Okay, and the second thing will be the which mutation results in the substitution for one nucleotide for another without changing the reading frame. Here, one nucleotide has been replaced by another nucleotide. Okay, and the reading frame does not change. Here, what is the type of that mutation? Is it a silent mutation? Yeah. See, when a nucleotide substitution does not change the amino acid sequence, there is a difference between a missense mutation and the silent mutation. Here, the protein, the amino acid does not change because there are different codons code for a same amino acid. Okay, here the codon has changed, but the protein code for that particular codon will not getting changed. Okay, in the missense mutation, there will be a different amino acid will be produced. Okay, there is a difference between here. And which type of mutation involves the substitution of purine nucleotide for another purine or a pyrimidine nucleotide for another pyrimidine? Here, purine will be substituted by another purine, and the pyrimidine will be substituted by the another pyrimidine. What is the type of substitution reaction mentioned here? That will be obviously a transition mutation. Which mutation type leads to a replacement of one nucleotide with a different nucleotide, potentially changing the amino acid sequence of a protein? Here, the amino acid sequence are getting changed. That will be the missense mutation. Okay, you don't want to confuse with the missense, nonsense, silent mutation and all. Okay. Okay. Which mutation involves the insertion or deletion of nucleotides in multiples other than 3, leading to the shift in the reading frame? Here, the shift has been changed in the reading frame. That results in the, that will be obviously a frame shift mutation. Okay, see, if any insertion or deletion happens and the reading frame has to be changed, okay, and that will be placed under the frame shift mutation. Okay, a substitution for a guanine for a thymine and would be the example for a mutation type. For that, you have to know which is purine and which is pyrimidine. Okay, see, for example, a purine will be the adenine and thymine. Sorry, adenine and guanine will be the purine, thymine and cytosine will be the pyrimidine. Here, one purine is substituted by a pyrimidine. So what is the type of mutation? That will be obviously a transversion mutation. Which mutation types involves the replacement of a codon with a premature stop codon, resulting in a termination of a protein synthesis? Here, the protein synthesis has been terminated. And what will be the type of mutation asked here? That will be obviously a nonsense mutation. Okay. What mutation type occurs for a single nucleotide is inserted or deleted, causing a shift in the reading frame? Once again, it will be the frame shift mutation. Okay. Just tell me which mutation type alters a codon to one that codes for a same amino acid, this not changing the protein sequence. Even though that will be a same question or repeating, just answer. It's just like a revision. Okay. That will be obviously a silent mutation. Okay. Okay, they're all same question. Okay, same thing is repeating. Okay, that's all about mutation. And do you have any doubts? Means you can ask me now, or we will wind up the class. For tomorrow, you have to read and come the chromosomal aberration, deletion, duplication, inversion, translocation, ploidy, and their genetic implication, and also there will be a chromosomal disorders. Okay, tomorrow the test will be from the 5th subunit, right? Everybody clear? Lethal is nothing but that mutation has to be definitely leads to a death of an organism. That is what a lethal. Okay, lethal means what? That mutation, if that mutation leads to a death of a particular organism, that has to be the lethal mutation. Okay. And in case of a conditional mutation, in only with respect to a particular environment, this mutation will happen. That is what a conditional mutation. Okay, biochemical means obviously that any metabolically, if that any mutation has occurred, that what a biochemical mutation, and loss of function and gain of function will be an important thing. And in case if the mutation happens in the germinal germ cells or the reproductive cell, that will be the germinal mutation. Okay, and if it is occurs in the body cell, that will be a somatic mutations. Right? Good. Okay. For tomorrow, just read and come the chromosomal disorders. Okay. Thank you.